Hello friends, this video will describe the basics of gene library. As we know, similar like to the book library, there is also one terminology which is very famous in a molecular biology, which is known as the gene library. Now let us understand what is the meaning of the gene library. There is one terminology that is called library. In terms of biology, it is nothing but it is the population of organisms which carries a DNA molecule, and this DNA molecule is inserted into a cloning vectors. That means it is somewhat similar like to the book library. As we know, whenever we are going to the book library, we see different types of the section in library. Like for example, there is a different section of the arts book, there are different section for the commerce book, and similar like to the science section, where you can easily access any of the book from the any of the section. Similar like here, this gene library is also the collection of DNA molecules which is inserted into the cloning vectors. So ultimately it is nothing but it is the collection of all the clone vector molecules, that is a gene library. In terms of molecular biology, a library is a collection of DNA fragments, and this DNA fragment which is produced from the whole genome of that particular organism, and this DNA fragment which is stored and propagated in a population of microorganisms through the process of molecular cloning. In the next slide, if you just see, gene library is of two types. gene library actually दो तरह की होती है, एक जिसको हम कहते हैं genomic library and another that is a cDNA library. So there are two types of gene libraries. One is called genomic library and another is complementary DNA library. At the end of this video, you will see what is the difference between genomic library and cDNA library. Now let us see. First start with the genomic library. Before we are going to the procedure of genomic library, we have to understand some basic phenomenon of, or either the terminology which is very popular in the molecular techniques. First things, whenever we are going to construct any of the gene library of any of the organisms, we must have to know what is the size of the gene that is produced after the fragmentation of the DNA. And another things that is most important is what is the capacity of the vectors. That means how much size of gene to be carried by any of the vectors. So there are two important things, size of the gene, and second one is the capacity of the vector. Now here the question is, what is vector? Vector is nothing but it is a one type of vehicle that will be carry the fragment of DNA gene. Another important terminology that we have to understand, that is the restriction enzyme. Now this is the most important enzymes and widely used in a molecular biology. The characteristics of the restriction enzyme is they have ability to cut at the particular nucleotide base, and that's why it is also popular as molecular scissors. That means they can able to cut at a specific site in the DNA segment. Another important enzyme which will be utilized to prepare the gene library is the DNA ligases. Ligases का काम क्या होता है? That will be join the two fragments of DNA. पहले हम कट करेंगे, बाद में हम जोड़ेंगे. Now another important things which will be utilized in the preparation of this gene library is vectors. There are different types of vectors which is generally utilized in the gene library construction. One is plasmid, another is lambda phase, cosmids, bacterial artificial chromosomes, and yeast artificial chromosomes. It is nothing but it is we can say the extra chromosomal parts of the different organism that is used as a vector to construct the genomic library. Now this slide shows what is the capacity of that particular different types of vectors. They can carry the DNA fragments. If you just see the plasmid carries only 20 kb kilo base pair of clone DNA. That means in this plasmid 20 kilo base pair DNA fragments, they can be inserted into the vector. Lambda phase, they also carry 20 kilo base pair DNA, cosmid 45 kilo base pair, and bacterial artificial chromosome, they can able to carry greater than 500 kilo base pair, and yeast artificial chromosome, they can ability to carry 1 mega base pair of gene segment. That means if you want to insert a small size DNA fragments into the vector, you have to utilize the plasmids. And if you have a larger DNA fragments greater than 500 kb or around 1 megabase pair, then you have to utilize yeast artificial chromosomes. Now before we are going to see the process of genomic library, we have to understand what is the genome. As we know, genome is nothing but it is the totality of DNA sequences. That means the whole genome, whole DNA genome, which includes not only the central DNA molecules but also the DNA which is present in the chloroplast and mitochondria in the case of plant cell and the animal cell. If you just see the smallest genome of the one of the plant, Arabidopsis thaliana, which carries 130 megabase pair genome, and for human the genome size is greater than 3300 megabase pair. Now let us see the procedure of how you can construct the genomic library. The genomic library can be constructed by extracting the whole DNA from that particular organisms. So the first step is organism's DNA is extracted first from the cell. Then after this isolated DNA is digested, that means cut with the restriction enzyme and produce a different types of DNA fragments with a specific size. You can here use different types of restriction enzymes which is available in molecular techniques. Once a DNA fragments is obtained, then this DNA fragments is inserted into a particular vectors that we have discussed earlier. You can use the plasmid vectors, cosmid vectors, lambda phase vectors. Okay. So this vectors in which you have to insert the different DNA fragments which is produced after the action of restriction endonuclease, and this can be proceed by the DNA ligases, because DNA ligases that will be join this DNA fragments with the vectors that you have to choose. The vector DNA, then after it is taken up by any of the host organisms, most popularly is E. coli or either the yeast, where this clone DNA can be expressed. Expression, that means we already know the central dogma theory, any of the DNA can be expressed and converted into protein only when they have a proper medium. So that we have to insert this clone DNA into the proper host organism like E. coli and yeast, where this clone DNA can be expressed and produce a proper amount of protein. So this is, we can say, the procedure of genomic library. This is just a diagram where you can understand how the genomic library can be constructed. For example, the very famous Drosophila melanogaster, that is the fruit fly. If you want to prepare a DNA library, then you have to first extract the DNA, whole genome of this Drosophila melanogasters. Then after, apply a specific restriction endonuclease and cut and produce DNA fragments. Similar, like side by side, you have to use a specific type of vector. For example, if you use the plasmid vectors, then you have to isolate the plasmid from the bacteria. This plasmid is cut by the specific restriction and endonuclease. So here the vectors will be open up. Now these vectors, in which each and every DNA fragments that is inserted into the vector and produce a recombinant DNA. Now this each and every recombinant DNA that is inserted into the host organism, where you can construct a genomic library. So this is a library. If you want a specific type of gene, then you have to select a proper clone from the library and express it. This is the same pictures where you understand how this plasmid will cut and DNA fragments will produce. This is the example where the plasmid is cut by the echo R1, that is the restriction endonuclease, and produce a sticky ends. Similar, like DNA whole genome, it is also cut by the echo R1 restriction endonuclease and produce a large number of DNA fragments with the sticky ends. With the help of the sticky ends, it will be hybridize this plasmid and DNA fragments and produce a recombinant DNA. With the help of DNA ligase, it will be joined to each other and produce a recombinant plasmid, which can be inserted into the host organisms. So this is about the this is about the genomic library. Now let us understand what is the cDNA libraries. It is also known as a complementary DNA libraries. A cDNA library represents a sample of mRNA which is purified from the particular source. Either you can use a particular types of cells, either you can use a tissue, or either entire organism, where you can extract the mRNA. Now it is, remember that cDNA library that will be represent an expression of the gene at the particular time point. So here, first step for the construction of cDNA library is the extraction of mRNA from the particular type of cell or tissue or organism, and this mRNA is converted back into the DNA by specific enzyme that is called reverse transcriptase. So here the enzyme reverse transcriptase is the most important part of to construct the cDNA library. There are various methods which is available for the mRNA extractions. One of the famous method, that is triazole extraction, or either you can use the column purification, where you can extract the mRNA from the particular type of cell. Once you extract the mRNA, this mRNA is to be converted into the DNA with the help of reverse transcriptase. As you know, this reverse transcriptase enzymes is generally present in the retroviruses. Now this retroviruses generally contains RNA as a genetic material which can be converted into DNA with the help of reverse transcriptase, and this DNA is inserted into the host genome once there is a virus infected to the particular cell. So this reverse transcriptase has been used to produce a DNA from the mRNA. So ultimately you have produced a clone DNA molecules that is engineered with the help of the various restriction and endonucleases. This is the chart that shows how the cDNA library is to be produced. For example, at any particular time point where one particular type of cells they express a particular genes which is in the form of mRNA. This mRNA is to be extracted from the tissue, and this mRNA is converted into the DNA with the help of reverse transcriptase. Now this DNA which is produced from the RNA is to be inserted in a particular type of vectors like either the plasmid vectors or the lambda phase vectors or either any of the vectors where you can insert this double-stranded DNA. Now this plasmid is to be inserted into the host organisms like bacteria, and in this way you can prepare a cDNA library. Now this slide that shows what is the difference between the genomic library and the cDNA library. Genomic DNA library is generally produced or constructed by the extraction of chromosomal DNA. The whole genome is to be extracted, whereas the cDNA library generally is prepared or constructed with the extraction of mRNA. Then after here, another difference is here the chromosomal DNA is digested with the restriction endonuclease, and each and every DNA fragments which is produced by the restriction endonuclease is inserted into the particular vectors that is considered as a recombinant DNA. This recombinant DNA is transformed into the bacteria, and in this way you can construct the genomic library. And cDNA library in which the mRNA is produced and converted into the complementary DNA by the reverse transcriptase, and this complementary DNA is inserted into the particular vectors that is called recombinant DNA, and the remaining procedure is the same like to the genomic library. Now here this slides that shows what is the difference between the genomic library and the cDNA library. You can see here this is the particular genomic DNA where there is two genes, gene 1 and gene 2. Now if you just see this black lines that shows the introns and the red color that shows the exons. As we know, exons that will be a codon sequences which ultimately produce the protein, whereas intron is the non-coding sequences. When you construct the genomic library, the whole genome is to be used. That's why genomic library that contains the promoters, introns, intergenics and non-expressed gene also. But the complementary cDNA library is generally produced from the mRNA. That's why it contains only the expressed gene, transcription start sites and open reading frames. This is the applications of genomic library in the different fields. You can also determine the complete genome sequence by preparing the genomic library. You can also use as a source of genomic sequence for the generation of transgenic animals. You can also study the function of regulatory sequences in vitro. You can also study the genetic mutation in the cancer tissue. Also, you can discover a novel genes, cloning of full-length complementary DNA molecules in vitro study for the gene function. You can also study the repertoires of the mRNA expressed in the different cells of the tissue and alternative splicing in the different cells of the tissue. So I hope you will utilize this video for your improvement of your knowledge. Thank you.